Okay, good afternoon. So I think I'll get started. So for those of you who don't know, I'm Chris Baker in the Laboratory of Brain and Cognition, the head of the section on learning and plasticity. What I'm going to talk about today is voodoo correlations and double dipping. And I'm going to explain what all those terms mean and what we're referring to in those particular cases. And during this talk, I have the slides here to sort of show you what I mean by these terms and why I think it's something we have to be very aware of when we're doing fMRI analysis. But I'm also happy to take questions on anything else related to this. So you'll see what's here is going to be sort of talking about how we do analysis and what we have to be careful of. But also in going through all of this, I've learned a lot about journal publishing, about how we do science communication. I may make some references to that kind of stuff as well, which I think is also kind of important to consider when we think about how we, within the field of, say, fMRI, discuss analyses and problems and errors in work and things like this. And it's actually kind of tricky to deal with. So what I'm going to talk about today, primarily you can find sort of covered in these three papers. So in 2009, myself, along with Nico Kriegskorter, Kyle Simmons, Pat Belgowan, published this paper, Circular Analysis in Systems Neuroscience. And this is our double-dipping paper. At the same time, in the same year, there's another paper called Puzzlingly High Correlations in fMRI Studies of Emotion, Personality, and Social Cognition. This is the Voodoo correlations. And the reason it was called that is because the original title of the paper was Voodoo Correlations. And so actually that changed during the review process, but that's kind of how people who were around at the time would think about this paper. And because it's now been 10 years since the original double-dipping paper, recently there was a short piece in Nature Neuroscience sort of revisiting this and discussing that as well. And I think in the context of what I'm going to present here, we can also talk about how any of the issues that I present are still things we need to be worried about today, and how we should think about it 10 years on. So now the best way to give you an idea of what I mean by double dipping or voodoo correlations is to really give you an example. And that's what I'm going to go through. I'm going to go through a number of different examples and sort of hopefully make it clear before discussing at sort of a more general level how to think about these things. But to make it sort of easy, there's a very simple take-home message from everything I'm going to present today. And that take-home message is that data selection, when you're selecting data that you're going to look at, that needs to be independent from any other data testing that you're doing. And so this is the take-home message. I'm going to put it up at the end, and hopefully it'll make more sense to you what I mean by that. But if you remember one thing, this is all you have to remember. Keep data selection separate from any other analyses that you're doing. So to start with kind of an example, and this is an example which is how I first sort of became aware of these issues and really got involved in kind of any of this discussion. And so just to give you a tiny piece of background, we'll just talk a little bit about faces. So when we put people, we can put somebody in the fMRI machine, and we can show them faces, a series of faces, a series of objects, and say, where do we find regions that are more responsive to faces than objects? And when you do this, here's just a lateral view and a ventral view of the brain. When we do this, we find a couple of regions. We find a region in lateral occipital cortex here that people have called the occipital face area, sometimes referred to as IOG faces using an anatomical reference. And this one on the ventral surface called the fusiform face area. So these two regions have been extremely well characterized. They're very reproducible that you can find these regions. But there's been this kind of big question, which is, are these really regions that are specialized for face processing? So there's a general selectivity advantage, but what exactly is going on in these regions? And also bearing in mind that, our resolution that we have with fMRI is still somewhat limited. So to address this question, in 2006, this paper came out, which was using high-resolution imaging to look within the fusiform face area and say, okay, is it really all face selective? And so what they did in this study is at 3 Tesla standard resolutions, they localized this fusiform face area using this contrast of faces versus objects. But then when they did sort of high-resolution scanning, now with much smaller voxels, and they looked within this area, what they found is that within this region, yes, you find regions, all these voxels that are highlighted in red here, that are most responsive to faces. But if you look at responses to animals, you can find some voxels within the FFA that are most responsive to animals, some that are most responsive to cars, and some that are most responsive to sculpture. These were abstract sculptures. And if you go through and you say, okay, well, let's look at these different types of voxels in detail, then what you see here is these face voxels have a strongest response to faces. This is F on the x-axis there, and much weaker responses to the animals, the cars, and the sculptures. If you go to the next set of voxels, you go to the animal voxels, now you find they have the strongest response to animals. The car ones have the strongest response to cars, and the sculpture ones have the strongest response to sculptures. So this, if you take this at face value, what this suggests is that within this region that's being defined as phase selective, there are voxels that are highly selective for different types of categories. Now, when this came out, this was of great interest to me because I was essentially running the same experiment in a different place. But this was not the result that I was finding. And so I spent a lot of time trying to work out what is going on here. This group was apparently finding these very, very highly selective voxels within the fusiform phase area that I was just not seeing with my own data. And what this sort of led to, after a while, I worked out what was going on. I'm going to take you through this. And this is going to be our first example. So it's sort of the example of what can happen with circular analyses or double dipping with univariate activation. So in the end, we ended up publishing a response in Nature Neuroscience to that article, which you can go ahead and you can read that, but I'll summarize it here. So what I'm doing here is putting up the data from the original study. So the first thing I wanted to do is say, okay, can we reproduce the data that they've got? So we ran the same analysis. And so this is now data that we collected. This is in the fusiform phase area region of interest. And now we break out responses to faces, animals, cars, and sculptures. And we can reproduce basically the pattern that they observed there. So this is great. We've reproduced their result. Sure, this is highly replicable, and everything is fine. The trouble is, there's a big problem with this analysis. And the problem with this analysis is that the voxels were selected based on their preferences, and then they looked at the activation of those in the same data set. So the same data is being used to select the data and then to look at the response profiles. So this is what we'd call a non-independent analysis. So selection and further testing with the same set of data. Now, the other way of doing this is to say, okay, we're going to take the voxels, label them according to face, animal, car, sculpture, and now, in a separate data set, we're going to get at the activation profiles of those in completely different testing. And if you do that, you do independent testing, then what we found in our data is a pattern like this. So this pattern here, as you can see, is incredibly different from what you see in non-dependent analysis. So now you'll see that for all the different sets of voxels, the ones that were most responsive to faces, animals, cars, or sculptures, when you look in independent data, now you see that all of them respond more to faces than to anything else. And so this really leads to a very, very different conclusion about what you might want to say about this fusiform phase area. So the basic problem here is that the data was sorted, first sorted into these different categories of voxels, and then now you extract the values. When it's the same data, you get the pattern on the left. When you look at independent data, you get the pattern on the right. Now, you can highlight this problem by looking a different way. So this is now looking within to the fusiform phase area ROI. But we did a second analysis just to highlight how this comes about by taking an ROI that wasn't in the brain. So we took an ROI that was actually in the nose, I think is where it actually was. And you can do the same analysis there. And what it's showing here on the left down here, this is the non-independent analysis. So you sort them according to their strongest response to faces, animals, cars, or sculptures. You pull out the responses, and look, they all have the strongest response to the category that they were originally responsive for. Of course they did. You selected them because they were most responsive to that category. But if you now take independent data, so this could be a separate run, it could be data quite on a separate day, what you now see is, in fact, you have no real responses to any of these categories and no real differences. So here, this kind of what we call the circular analysis, double dipping, so selecting in the data and then going back to investigate what the profile response is like, can lead to this kind of spurious result. And so here, the point here is this is an example of double dipping where it can produce an effect where there's no effect at all. Right? And you could do this analysis, we did it in a non-brain ROI, you could generate random data. And if you do the selection and pick, simulated voxels because they have the strongest response to one category, and then in that same data that you generated, now plot them, you'll find the strongest response to the category you selected them for. So this is obviously a problem. And so here there's just, one of the things you might think here is, well, okay, this is one example of this. So where else can we have an effect of double dipping? So the thing is, even if you have an underlying effect, you can still distort the data by doing these kind of analyses. So let me give you an example of that. And this one's going to be based on a simulation. So here what we're going to do is we're going to simulate a bunch of voxels. So we're going to take an arbitrary 3D volume of voxels, and we're going to put an effect in some of those voxels. And we're going to effectively run like a block design experiment. We have 4 conditions, A, B, C, and D. And in this case, we add some noise to the underlying data. And this is just sort of Gaussian noise that's been slightly smoothed. And so we can take a slice from our simulated data. This is a central slice here. And we put an effect in this region, this set of voxels here. And the effect we put in is a strong response to conditions A and B, but no response to conditions C and D. So this is the true underlying effect. But now what we do is we go through, and we're now going to say, OK, within our voxel volume, we're going to search for voxels, having simulated this set of data, we're going to search for voxels that now have a stronger response to condition A than condition D. So we have this sort of contrast we're testing. This is our contrast hypothesis, A versus D. And we do that, and we're going to find this set of voxels here. So now these are the voxels that now show a stronger response to A than D. And if we do kind of the double dipping, and we say, OK, these voxels show A than D, now I'm going to plot what those voxels did, then you'll find something that looks like this. So here what you can see is these voxels all respond more to A than to D, but now you can also see they respond more to A than to B. And so this is true, and this is very important, this is true even though when we select those voxels, we're selecting them according to kind of appropriate metrics of multiple comparison correction, all these kind of things. It's not about multiple comparison correction. It's about using the same data to select voxels as then to look at your effects. So if we now go through and do a different way and say, OK, now we're going to define our ROI using independent data. So here's now an independently defined ROI in this simulated data. And now you pull out of that independent data what the results are. You can see that actually you end up with something that looks like what the true effects were. And one way to think about this is that what we have here is we have the ground truth, which in this case we knew what the ground truth was because we simulated the data. We have our hypothesis. And when you run this non-independent analysis, what you're actually doing is you're kind of blending the underlying effect with whatever the hypothesis is. And depending on depending on the nature of the noise and depending on a number of different factors, you'll end up with somewhere between these two distinctions where you get your same data. But of course what we want to do is we want to try and find what this underlying ground truth is, and so we use independent data to do this testing. So does this make sense? I mean, so the main thing that I'm harping on here is this idea that selection needs to be independent from whatever test you're later performing. So here, in this particular case here, this is now defining the ROI and then extracting the response. In the first example, that was sorting the data based on some property and then looking at kind of the response profiles. So in this simulation here, what you can see is that this double dipping can produce a distortion even when there is an underlying effect. And it doesn't matter how strong your effect is, it doesn't matter whether you use multiple comparisons, you're still going to have this kind of issue going on to some extent. So let's go with another example. So now we've looked at kind of like univariate kind of activation style analyses. This is also a problem if you're going to do some sort of multivariate analysis. So here we're going to look at like a multivariate pattern analysis, or MVPA. And this is an example that comes from work that was done at NIH. So Kyle Simmons in Alex Martin's lab and people had run an experiment where they had 2 different factors. They had a stimulus factor, which was object category. And so in this case, for example, they had faces and dogs. And they also had a task. And the task was to make some sort of property judgment. So people are seeing either the faces or the animals. And they're making a judgment, which is either, is it animate or inanimate? And is it pleasant or unpleasant? And what they did is for each of these kind of combinations, so for each stimulus category and each task, we can look at the patterns of response across this ventral temporal region of cortex. The type of analysis they did here is actually one that was based on the work of Jim Haxby from 2001. They did this split half analysis, where what you do is you take half the data, take it into 2 halves, and then you look at the correlations in the patterns of response across those 2 halves of the data. So first of all, what you do is you define your ROI. And they did this by just selecting voxels for which any pairwise condition contrast among all the different conditions was significant. Then they ran this correlation analysis, split the data in half, and you can think about those 2 halves as being like a training and test set, although it's kind of symmetrical in this particular case. And now you compare the activity pattern for same condition versus different condition. If you have a stronger correlation when it's the same condition across the 2 halves of the data than when it's different conditions, then you have some ability to decode from the pattern of response which condition is present. So here's the data that they got. So we now have we've turned this into a decoding accuracy. And now what they found is that for both task and stimulus, you can determine what was going on at that particular time. So this is great. This is what they were looking for. They were looking for a strong effective task, and they found it. Now this is in the fMRI data. Now all was well and good until they decided to run a control analysis where they took randomly generated data. So they took randomly generated data, ran it through their analysis pipeline, and what they found is that in this randomly generated data where there was no effect, they could decode both task and stimulus. So the question in this case is like, okay, what's actually going on here? Because in this particular case, the two halves of the data, we had independent halves of data that were being tested for the correlation. The problem here is a little bit more subtle. So the problem here was that when they selected the voxels, they used all of the data. So across both halves, they used the data. And when you do that, what's going to happen is, imagine you've got some sort of noise in your data. You're going to select voxels across all of the data, and that's going to tend to bias you to voxels that showed similar patterns, similar responses in the first and the second half. And so what you've done is you've now limited yourself to those voxels that already show some consistency before you now do your correlation across the split halves. So the other way of doing this is say, okay, well, let's not use all of the data to select the voxels. Let's just take half the data. So say you've got your odd and even runs of your data. You take the odd runs, you use that to select the data, and then now you look at the correlation across the two halves. If you do that now in the fMRI data, what you find here is that you get an effect for stimulus, you get no effect for task. If you do this in the randomly generated data, you get no effects at all, which is kind of what you'd expect to do. So here, the problem is not so much this sort of replotting the data on the basis of selection. The problem here is how they defined the region of interest within which they did the analysis. And again, they used the same data to select the voxels, because they were using all the data to select the voxels, that later was coming into the comparison in the split half. And so in this case here, what this sort of shows us is that the test data, the data that we're actually using, you shouldn't use it in either training your classifier or whatever decoding analysis you're using, but you also shouldn't use it in defining the region of interest. And so again, keep the data separate from the different parts of the analysis. So now we can go to a third example. And now this is the one that kind of where the Voodoo correlations paper was coming in. So the Voodoo correlations paper was particularly focused on brain behavior correlations. And their big concern in that paper was that they were seeing papers that would say, okay, within this region of the brain, we're seeing a correlation between brain and some behavioral metric with an R value of 0.8, 0.9. And their feeling was that this was just too high to be believed. That actually, given the reliability of these metrics, given the reliability of brain imaging data, these correlations just seemed like they could not be that high. And that was why they focused. And in the end, what's sort of underlying what they were pointing at in their paper is the same kind of issue of non-independence of selection and test. So the example I'm going to focus on here is one that came from Russ Poldrak's lab. So here, in this particular paper here, they were looking at loss aversion in decision making. Now, I'm not going to go into all the details of this study, but all you need to know is that they had some measure, based on the fMRI data, of loss aversion. So people's reluctance to lose money or something in some sort of gambling task. So what they did is to say, as part of this paper, is to say, okay, let's find brain regions where we get this correlation between whatever's happening in that brain region and sort of people's behavioral loss aversion. And then when they found those regions, they pull out like the correlation. So here what plotting on the x-axis is this measure of the neural loss aversion against the behavioral loss aversion. This is from somewhere in the striatum. And so what they find is that when you run your whole brain analysis, you find the correlation in this area. If you now look at your data, you get this kind of beautiful correlation here. You get an R value of 0.85, you get a P value of less than 0.001. But, and this is not the only region where they found it. Here's a listing of all the separate regions where they found these kind of correlations. But the problem here again is this one of independence. This was a non-independent analysis. So if you search throughout the entire brain and find those voxels that show a correlation, and now you find, okay, this one shows a correlation, now I'm going to plot what its data is. Well, of course, you've pre-guaranteed you're going to have a correlation because that's why you selected the voxels. But more than that, what you're doing is you're inflating those estimates that you've got because you've got noise in your data as well. And so now basically you maybe have cases where the noise just happens to agree with whatever effect is going on. That's going to add to whatever your underlying effect is. So to give you an idea of what the magnitude of this is, So Poldrack actually, after the Voodoo correlations paper came out, they went back to this study and they decided to run an independent analysis, say, well, how bad is this for this paper? Is it the case that if you now look at this again, that all these effects are just going to go away completely? And that's not the case. So they ran the second follow-up paper here, this one, independent scenario analysis, whereas the Voodoo, and they went through and they now ran and they compared independent versus non-independent analyses. And so if we go back to this same set of regions here, what I'm showing here is where it says this is the sort of number of voxels in each of those regions. The test R is when they had independent data. So they search for the voxels with one set of data, and now in other data, they're now going to look at the brain behavior correlation. And the one on the right is when they use the same data for searching and for plotting the correlation. And you can look across all these and you can compare those R values. What you really want to focus on here is this estimate down here, this bias value. So that's the average difference in the R value that they observed between doing the independent versus non-independent analysis. And so in this case, it's not the case that these correlations completely disappear, but the magnitude of those correlations does go down substantially. So here what we're seeing is that double dipping in this particular case is inflating these brain behavior correlations. What's actually happening is you're getting a larger effect size than probably what the true underlying effect really is. So to sort of step back from this and kind of talk more generally about what's going on here, we can sort of think about what we're actually doing with an analysis. So when we have an analysis, we have our data, and the way we often think about it is we go from our data, we look at our data, we go all the way across in some kind of like nice little walk, and we get our results, and that's what we publish in our paper. The problem is that between the data and the results, we have this thing that is the analysis. And again, we may look at our analysis and think, like, this was so logical, everything was so clear, what I was doing here. And you think you've got this nice sort of straightforward path from data to results through your analysis. But, increasingly, given the nature of the data we're getting, given the size of the data, things may actually be a lot more complicated. So actually, analysis isn't often sort of straightforward path from data to results. This is kind of okay. You can lay everything out, and this might not be a problem. But you've also got these things which are the assumptions. So whatever analysis you're doing, you have a set of assumptions. And ideally, those assumptions go into the analysis, but it's not really going to affect how you get from data to results. So we'd like to think that the data goes in, you combine it with some sort of assumptions, the results come out, but still it's kind of like there's like a strong connection between the data and the results. The trouble is that's not always the case. The worst case scenario is going to be that the data goes in, you have your assumptions, your analysis, and what comes out is actually directly resulting from the assumptions that went in. So the data sorting example that I showed you at the beginning is kind of like that. An effect was produced that was not there without the assumptions that underlie the analysis. And in this particular case here, you can think there's a direct path from assumptions to results. Now, that's why we call this circular inference, right? So basically, the assumptions you're putting into your analysis directly produce the results that you're ultimately reporting. Now, this probably isn't the case in most examples that you might see of this. More likely is that what you get is the result of some sort of combination of the data plus the assumptions that you put in. And so we need to understand kind of exactly how these kind of assumptions fill into the data. And I've sort of given you some different ways in which you can look at this. Simulating your data is one way you can try and get a sense of like, okay, are my assumptions sort of producing the results that I'm actually reporting? So why exactly then do the assumptions that we have underlying fMRI analyses, or often have underlying fMRI analyses, produce this circularity? So I've already talked about this, but I just want to sort of go through it in this sort of more abstract form. So what I'm highlighting here is the basic problem is often this case of selection, of selecting the voxels. In fMRI, we always have huge amounts of data. We have hundreds of thousands of voxels, and most of the time there's this selection process or this search across the entire brain that's going on in the data. Now, selective analysis in and of itself isn't a problem. Selective analysis is kind of very useful and can be very powerful, right? This often we need to be able to do this to actually make any sense of our data. And this is partly because of the size of the data sets that we're doing, and it's also important to make sense here that this is not just about fMRI. This is common throughout neuroscience. We have very large data sets. Data selection is often something that's happening in most of the data that's out there. So the bottom line about why selection can produce this circularity is that it's when the result statistics, so whatever kind of analysis you're doing on the data, if they're not independent of the criteria used for selection, then you have this circularity. And that's what I'm showing in these examples. And the reason why is because when you're selecting, we're not just selecting based on the true effect. In the data, we have our true effect, and this is combined with all various sorts of noise. So we're selecting on the basis of the true effect plus noise. If our data was noise-free, this wouldn't actually really be a problem. Because we have the noise, and the noise is changing whatever underlying true effect is there. Sometimes that noise will happen to agree with whatever hypothesis. Sometimes it will tend to disagree with it is. Sometimes it will sort of change the level of an effect that we see there. When you go through and do selection, you're selecting the noise that happens to agree with your hypothesis. It's effectively what's happening. And there's many different types of selection. So here, you can think about binary selection. You either select or don't select. You can actually do some sort of weighting selection where you look across different sets of voxels and different sets of data, and now you're going to weight how strongly you want to look at that data. You have sorting, which is the first example that I gave you. All of these are just different types of selection that can go on. And in these cases, you need to be careful to make sure that the selection is independent of whatever test statistic you're going to run. Now, of course, this can be kind of easy. It's like there's ways to avoid this problem are, one, we talked about separate data sets. You can also think about separate data sets as being, well, say, for example, you have an anatomical region of interest, and now you're testing some effect in functional MRI data. That's going to be fine, as long as you're not using the functional MRI data itself to determine that kind of selection. So the examples of selection, we talked about, in fMRI, we have the region of interest or the voxels of interest. But I want to sort of emphasize again, because the whole context of this debate was actually around fMRI. But this is not just a problem for fMRI. And this can come up most of the time. You're looking at other types of data. It's also a problem in other types of data. So we can also talk about how it can show up in other domains. So let's think we're doing physiology. We're doing single unit recording from neurons in the macaque brain, say. So now one example of sort of circularity is going to be the case of we have our paper, we want to illustrate what the effect is, and we're going to show a typical neuron in the paper. This is a very common thing to do. That in itself is kind of there's a selection process in there that can mislead about the strength of the effect. We also can, you can pre-specify selectivity. So you might go in and record, and now you say, okay, we're only going to take neurons that are visually responsive. Now that may be important because you're only interested in visual responses, but what you have to recognize is that is also going to have an effect on what you might observe. And there's ways in which how you define that selectivity that can cause a problem. Another example might be, say, let's say you go in and you record, and we could say an example in, say, I'm looking at numerosity, presenting different numbers of dots on the screen, and say recording response of a neuron to whether you have 2, 3, 4, 5 dots. Collected all this data, I go through now and say, okay, look, some neurons respond most to 2 dots, some respond to 4, some respond to 3. You now go through and say, I'm interested in what their tuning curves are like, so I'm going to take all those neurons that responded most to 2, average their data together, and get an estimate of that tuning curve. Or you could be 5 or whatever. That in itself is now a circular analysis. And if you went through, if you just had noise data, and again, if you selected them and said, okay, where do I get the strongest response to what would be the 2-dot condition, if you only take those ones that showed that strongest response, of course they're going to show the strongest response. So there's ways in which this can affect other types of data as well. Don't think this is just an fMRI problem. We could have this in EEG or MEG, where you might be selecting sensors or electrodes of interest. Or maybe you're selecting waveforms of interest. You want to look at the N170 or some other kind of feature of EEG activity. That selection process can actually bias what you now observe in the later parts of your analysis. In behavior, sometimes what people do is they have run a whole bunch of different subjects, and now you want to do some analysis where you say you want to look at, fast performers versus slow performers, fast learners, slow learners, something like that. And now, so you've got their data, and you're now going to bin people according to what their performance was like. Now, that can be okay, depending on what you're going to look at, but sometimes, and I've seen this in papers before, where they do that binning and now test and show, oh, when we bin them according to fast versus slow, there's a significant difference between them. And of course there is, because that's how they were selected. So it can come up in all kind of very simple ways. It can come up in genetic analyses, where you now maybe selecting subsets of genes. So the bottom line here is any kind of analysis where you have a selection of data from a larger pool before you look at it can cause some sort of bias in the effects that you now observe in your analysis. So, at the time that we were looking at this was kind of in like 2007, 2008. And, we sort of realized that this was actually quite common in the community based on our own reading the literature. And what we decided to do was actually say, well, okay, how common is it? Let's actually do a survey and see what do we actually see in the literature. So what we did was to say, okay, well, let's just take, this is for the year 2008, the whole calendar year. We took all the fMRI articles published in Nature, Science, Nature Neuroscience, Neuron, and Journal of Neuroscience, and went through them all. And went through all the figures and had a look at them and tried to work out, okay, how many of these papers that have been published actually contain what is a circular analysis. So out of, at that time in 2008, 134 papers in those journals, the number will probably be a lot higher now, we found that 42% of them contained a non-independent selective analysis, so some form where they've selected the data and done some later test using the same data set. On top of that, there was an extra 14% where we just couldn't tell because there wasn't sufficient methodological detail. And this is kind of another important message that sort of comes about through all of this, is the importance of being clear about our methods. People should actually be able to know what we've done so they can see whether there's a problem or not. And so overall, I mean, you can see here, this is actually more than half the papers where we would question the nature of some of those analyses. But the one thing that I want to make clear here is this doesn't mean that all these papers are wrong. Right? This is not a case. This sort of highlights that this was a common error. The groups that have done this and published stuff, they weren't alone. There were lots of other people doing this. And the thing is that even though these analyses may not be correct, we don't know if there's a true underlying effect there or not. It might be they've inflated the underlying effect, or it could be they've created one where there was none at all. And it's really hard to know unless you go through and, say, have independent data where you can necessarily test this. Or go ahead and run some sort of sophisticated simulation where you can try and test for that particular data would there be a bias be introduced there. In the case that I showed you at the beginning, I had independent data that was basically the same experiment, and I could actually go through and see, okay, if we do the same analysis, can we show that there's this problem? That's not always possible with what's there. So it's not the case that when you go back to the older literature, you should just ignore anything where I do analysis like this. The problem is that we just don't know how large that affects it, and then it's going to be important to sort of replicate those findings with future work. But here's the question, today. So that was 2008, so that's 11 years ago. So today, how common is it that you find these kind of non-independent analyses? And so for the article that was written a few months ago by Catherine Button, sort of looking 10 years on from circular analysis. So in that article, she kind of suggests that actually maybe it's still pretty prevalent. My own take on it is in reading the literature, I don't see it as obvious as it was at that time. When we went through and surveyed the literature, it was very, very obvious that there were problems in some of these analyses. We don't see it anymore. Now, it's possible people have just become better at hiding it. Or it's possible that we really had some impact in publishing this paper and people kind of paid attention to it. I don't think it's the case that it's not there at all. From working as an editor and a reviewer, yes, it definitely comes up in review. I've seen this raised a lot of times in review still. So it's still going on. So this is something you should still be paying attention to. And when you look at analyses, I think one of the problems that we have today is that the analyses have actually tend to get much more complicated. And also sometimes harder to see exactly what's going on. And I think it just means that you have to sit down and think through very carefully what's the basic logic of the analyses that people are doing. I'm actually pretty optimistic about it. I think that this is not as common, but it's something that you should definitely be aware of. So I'm going to put back up the take-home message, which is data selection should be independent from any other data testing. And that's the one thing you have to remember, and think about all your analyses. Now, when we published our original paper, we had a lot of people saying, oh, well, can I do this? Can I do that? Maybe if I do it this way, there's only a slight bias. Is that going to be okay? People complaining because we were sort of saying sometimes if you publish your data, if you have your data and you have your analysis and say you run a correlation, sometimes they just want to look at the correlation plot to look for outliers. And so some people got upset and we had comments about, can we not even look at our data? And the problem is, and I've generally taken a hard line on this, is that, yeah, it can be the case. The selection will only produce a small bias in the data. But the safest thing to do is, unless you've got a good way of estimating how strong that bias is and how much you're affecting the data, just keep it independent. And what we've tended to advocate, and what I still focus on today, is the idea of just keeping, if you have one data set, you can split that data. And that's a way of keeping having separate selection and testing. The complaint there could be that you've lost a lot of power by splitting your data. My answer to that is, well, if you don't have enough power to split your data and check the size of your effect, you don't have enough power to be estimating your effect size anyway. And you can't then decide to go with analysis that may actually be biased in the results. So I'm generally take a hard line on this. And even if you think there might not be a problem, if you can find a way to keep things separate, it's going to be much, much better and much safer. So these are all the slides that I have today. But I'm very happy to take questions on any aspect of this. And if you do ask questions, if you use the microphone, then it will be recorded for the recording. Okay, thank you. Any questions?
Do you think this is worth thinking about for resting state analyses where people find correlations in the data and some of that could be spurious, right? And do you think those analyses should also be split in half or done at multiple time points?
So I think the question there is, I think it's okay to say do an analysis. You can do a resting state analysis and you can look for an effect and you can say, I found that effect in this particular region. And the problem comes in is when you want to estimate what's the effect size. So I think if you run the analysis, you can be confident that, yes, I have an effect. If you can do all your multiple comparisons correct and all these things, you can say, yes, there is an effect there. But yet, to get at the size of that effect, that's where you need the independent data to say how much does it go there. Because in general, you're going to find that in independent data, the effect is going to go down. And if you want to have the unbiased estimate, that's what you need to do.
And then just to play devil's advocate in terms of effect size, it's generally good to visualize one's data and to see the effect, right? But I guess your point is that you can maybe show that data, show the effect, but not necessarily report it as an effect size or as an independent
So
sample.
you can use it as a visualization, you can use it as like a quality check or maybe, but you've got to be careful that plot in itself you shouldn't be using for inference, direct inference. And so one way of doing that, I mean, so one thing can come up is say for example, you run an experiment, I don't know, you run some sort of multi-way ANOVA analysis across different factors in your whole brain, and you find some type of interaction effect, and that's what you're looking for. But what you may want to know is you want to know what is the direction of that effect, right? You don't want to know just as an interaction. Maybe you really care about which is the nature of that interaction effect. And so then you want to look at the data because that'll tell you what's the nature of that effect. The key thing is it's okay to look at that data and sort of interpret the direction of your interaction effect, but again, not the magnitude of the differences that you observe there. And so in those cases, what, for example, we do in my lab and what other people start to do is you can plot those, but when you plot them, don't put on things like error bars and stuff like that. Because the error bars themselves are actually misleading. Those error bars are now biased estimates already. So you should think about having a purely descriptive plot that goes along with that. And you can include the statement and say, this plot is for visualization, it's not for inference. And it may seem silly to do that, but I think it's kind of important to be clear on where exactly is it coming from. Because even though some people may say, oh yeah, I put that plot up there, I know that I'm not using that for inference, I'm just saying to visualize it. If you don't make it clear that people should not overinterpret that, you're still sort of misleading by showing this thing that looks really nice. In the same way as like the single neuron example, right? I mean, on the one hand, it helps illustrate your effect, but yet, unless you randomly sample that, you're kind of giving a misleading impression about the strength of the underlying effect. Okay, anything else? Okay, well, thanks very much.